Welcome to Computational Biology. Today, we're going to explore metabolic pathway databases. If you have ever wondered how scientists make sense of the millions of chemical reactions happening inside a living cell, you are in the right place. We will learn how to turn complex biology into simple mathematics, step by step. Let us start by imagining a bustling city with hundreds of factories working day and night. In this city, factories take in raw materials, process them, and pass them along to build a final product, like a car. A living cell works exactly the same way. Molecules are the raw materials, enzymes act as the factories, and the chemical reactions are the roads connecting them. We call these interconnected cellular roads metabolic pathways. Understanding these pathways is the absolute first step to understanding life at a molecular level. Now that we understand what a metabolic pathway is, how do we keep track of them all? This is where metabolic pathway databases come in. Think of them as highly detailed, curated digital maps for a cell. They store all the routes, intersections, and traffic rules of cellular chemistry. Prominent examples include KEGG, EcoPsych for E. coli, and Metapsych. Instead of just listing parts like a dictionary, these databases map out dynamic interactions, allowing researchers to query, visualize, and simulate an organism's entire metabolism. These databases are incredibly powerful, but there is a catch. To make a computer understand a biological pathway, we cannot just show it a picture of chemical structures. We must translate the biology into a language computers understand, which is mathematics. We do this using graph theory. By representing a metabolic pathway as a mathematical graph, we can use powerful algorithms to search the database, analyze the network, and find critical enzymes. This transforms biology from a descriptive science into a predictive, computational science. This brings us to our first mathematical translation. We represent the entire metabolic network using a simple formula. G equals V, E. Conceptually, this defines the whole system. The symbol G represents the entire metabolic graph stored in the database. The variable V stands for the set of vertices, or nodes, which represent the chemical compounds and metabolites. The variable E stands for the set of edges, or links, representing the chemical reactions and enzymes connecting them. If the number of vertices increases, the network becomes exponentially more complex to analyze. Let us see this with a concrete example, using a tiny piece of glycolysis found in almost every database. We have three chemicals: glucose, glucose 6-phosphate, and fructose 6-phosphate. First, we assign a mathematical vertex to each compound. V1 is glucose, V2 is glucose 6-phosphate, and V3 is fructose 6-phosphate. Our final set of vertices, V, contains these three distinct nodes. This tells the computer exactly how many chemical entities exist in our mini network, forming the foundational list of items for our database query. Now that we have our vertices, we need to connect them. The first reaction uses the enzyme hexokinase to convert glucose to glucose 6-phosphate, so we create an edge connecting V1 to V2. The second reaction uses phosphoglucose isomerase, creating an edge from V2 to V3. Our final set of edges, E, contains these two pairs. These edges represent the actual chemical reactions that link our database entries together into a functional pathway that the computer can now process. Now that we have built our graph, we can analyze it. A key metric is the node degree, which tells us how many reactions a metabolite is involved in. The formula is K sub I equals the sum of A sub ij from j equals 1 to n. Here, K sub I is the degree of the specific node. A sub ij comes from an adjacency matrix, which is 1 if there is a connection and 0 if not. N is the total number of nodes. A high degree means the metabolite is a massive hub, while a low degree means it might be a dead end. Let us apply this formula to our glycolysis example. We want to find the degree for our second node, glucose 6-phosphate. We look at its connections. It has one incoming connection from the first node, glucose. It also has one outgoing connection to the third node, fructose 6-phosphate. Adding these together, 1 plus 1, gives us a final answer of 2. Physically, this means glucose 6-phosphate acts as an intermediate bridge in this specific pathway. It is neither a starting point nor a final dead end. To make this completely clear, let us look at a visual representation of what we just calculated. On the left, you see the mathematical graph. The circles are our vertices, V1, V2, and V3. The arrows are our edges, representing the enzymes hexokinase and phosphoglucose isomerase. On the right is the adjacency matrix. Notice how the grid uses 1s to show where connections exist and 0s where they do not. This matrix is exactly how the database stores the visual graph in a computer's memory. This mathematical mapping leads us to a very exciting application: drug discovery. When researchers look at a pathogen's metabolic database, they search for choke points. A choke point is a critical bottleneck in the network where a specific metabolite is produced and consumed by exactly one route. Because there are absolutely no alternative routes, blocking this path kills the pathogen. This allows scientists to design highly targeted drugs that destroy the bacteria without harming the human patient, turning data into life-saving medicine. To find these choke points computationally, we use specific degree conditions. The formulas are simple. K sub I equals 1 and K sub O equals 1. The variable K sub I represents the input degree, which is the number of reactions producing the target compound. The variable K sub O represents the output degree, which is the number of reactions consuming it. When both are exactly 1, it means the cell is entirely dependent on this single linear sequence of events. If either variable increases, the cell has alternative survival routes. Let us evaluate a pathway in a deadly bacterium using these conditions. The database reveals a specific metabolite produced by exactly one enzyme, meaning our input degree, K sub I, is exactly 1. This same compound is consumed by exactly one other enzyme to make an essential survival protein, meaning our output degree, K sub O, is also exactly 1. Because there are no other edges connecting to this compound in the entire database, our conditions are perfectly met, making this a prime target. Now, we must mathematically prove that this compound is a true bottleneck. We define a new variable, R, representing the number of alternative routes available to the cell. We calculate R by subtracting the primary route, which is 1, from the total incoming routes, K sub I. Since K sub I is 1, we get 1 minus 1, which equals 0. We have proven that there are exactly 0 backup pathways. If we design a drug to block the first enzyme, the bacterium will inevitably die. Proving this is a perfect drug target. We have covered a lot of ground today. Let us summarize the most important ideas. First, metabolic pathway databases like KEGG and EcoPsych act as digital maps of cellular chemistry. Second, we use graph theory to translate these biological networks into mathematical formats that computers can analyze. Finally, by calculating node degrees and finding choke points, we can identify critical vulnerabilities in pathogens, opening the door to advanced drug discovery. You now understand the foundation of computational biology and how it saves lives.